Usually when we talk about a medical diagnosis, there's this expectation of mechanical precision.
Right, like it's just a simple cause and effect.
Exactly. Like you break your arm, the X-ray shows that jagged white line, and the doctor just points and says, well, there it is.
Yeah, easy to see, easy to fix.
But I want you, our listener, our favorite lifelong learner, to imagine having a deeply inflamed, intensely painful organ hidden away in your abdomen.
Oh, man.
Yeah, doctors can't easily reach it. They can't just take a biopsy without risking, massive complications.
Right. The pancreas is notoriously touchy. You poke it and it gets incredibly angry.
Exactly. So you're left treating the symptoms, but never really understanding the root cause inside your specific cells. And that is the reality for patients with chronic pancreatitis.
It really is. It's the absolute definition of diagnostic muddy waters.
But we are looking at a groundbreaking August 2026 paper today from Cell Stem Cell.
Such a fantastic paper.
It really is. It's by Osorio-Vasquez and their team, and it completely flips this diagnostic nightmare on its head.
It does. They basically figured out how to grow personalized avatars of a failing pancreas in a petri dish.
Avatars, like biological twins they could safely crash test cures on. Okay, let's unpack this. Our mission today is to figure out how they built this biobank of patient-derived organoids, or PDOs, and what it means for decoding the disease.
Right, and it's a massive leap forward. I mean, we're moving from guessing what's driving the disease based on external symptoms to actually watching the disease operate in real time.
Which is wild. So let's get right into the biological mechanics of this biobank.
Sure.
Because culturing primary tissue, especially from a highly inflamed digestive organ like the pancreas, is notoriously difficult.
Oh, incredibly difficult. The enzymes alone make it a nightmare.
So how many of these avatars did they actually manage to establish?
Well, they attempted to establish organoid lines from 51 different patient samples.
Okay, 51.
Yeah, and these were patients actively undergoing surgery for chronic pancreatitis. And out of those 51 attempts, they successfully established 37 PDOs.
Wow, 37. That actually sounds pretty high for such a difficult tissue.
It is. In the world of primary cell culture, a biobank of 37 living, functionally robust avatars, for a disease this complex, it's a monumental achievement.
But they did lose 14.
Right. The remaining 14 samples simply couldn't be sustained, which really just speaks to just how challenging this tissue is to work with.
Yeah, that makes sense. And the demographics of those 37 patients, they tell a really compelling story about what actually drives this disease.
They really do. It totally challenges some common public assumptions.
Right, because if you look at the breakdown, out of the 37 successful avatars, 16 of them, so about 43%, were driven by genetics.
Nearly half.
Yeah. And another 13 patients, or 35%, were categorized as idiopathic.
Meaning doctors basically had absolutely no idea what caused their pancreatitis.
Exactly. And then, surprisingly, only 3 patients, about 8%, had alcohol-induced pancreatitis.
Which is usually what people think of first when they hear about pancreas issues.
Right, exactly. And the rest were just a mix of really rare cases, like 1 physical obstruction, 1 groove pancreatitis, and 1 solid pseudopapillary neoplasm.
That breakdown is so critical. It highlights how heavily this disease leans on molecular and genetic anomalies rather than just, environmental factors like alcohol.
Yeah, it really shifts the perspective.
And if we zero in on that genetic group, that 43%, the researchers tracked highly specific mutations.
Like CFTR.
Exactly. Yeah. CFTR mutations accounted for 27% of the patients. They also found PRSS1 mutations in 19% and SPINK1 mutations in 16%.
And just to clarify for everyone, those are the genes responsible for managing digestive enzymes and fluid transport, right?
You got it. When those genes fail, the pancreas essentially starts digesting itself.
Yikes. So to help keep those primary drivers straight, I was thinking about the acronym GIA.
Oh, GIA. I like that.
Yeah, genetic at 43%, idiopathic at 35%, and alcohol at 8%. It's fascinating that the unknowns and the genetics vastly outpace alcohol.
It really is.
But wait, before we get into testing these organoids, how do we know we aren't just looking at random scar tissue?
Ah, the validation phase.
Right, because the pancreas is an incredibly complex microenvironment. It's got exocrine cells producing digestive juice, endocrine islets making insulin, immune cells, fibroblasts.
It's a crowded neighborhood, yeah.
So if we just take a chunk of a diseased pancreas and culture it, how did they prove their avatars were actually representing the diseased ductal epithelial cells?
Well, that was the first major hurdle. They had to rigorously validate the identity of their organoids before they could even think about running any functional assays.
So what did they do?
They used expensive molecular profiling and mapped the expression data onto a heat map. And what they saw was incredibly high expression for specific ductal markers.
Like which genes?
Oh. Genes like KRT19, CA9, SLC9A3R1, and SOX9. Those are the definitive chemical signatures of ductal epithelial cells.
Okay, so they confirmed the presence of the right cells. But did they confirm the absence of the wrong ones?
Exactly. That is the other half of the validation. The expression data showed a total absence of acinar markers like AMY2B and CPA2.
Okay, good.
And it was completely negative for islet markers too, like GCG and INS for insulin.
Oh, wow. So no insulin-producing cells at all?
None. They also found zero expression of PTPRC, which marks immune cells, and absolutely no markers for fibroblasts or endothelial cells.
So they essentially mathematically proved they had washed away all the noise of the immune system and the stroma.
Right. They isolated only the pure diseased ductal epithelial cells.
Okay, so let's do a quick mini recap here. We are about 5 minutes in.
Good idea.
Bullet 1, we've got 37 successful chronic pancreatitis organoids created from diverse patient backgrounds. Bullet 2, the highest causes were genetic and idiopathic. And bullet 3, molecular markers prove these are pure ductal epithelial models.
Perfectly summarized.
So I have a question for you, the listener. Think about your own family's medical history. If scientists could grow an avatar of your cells, would you want them to test for genetic anomalies or unknown idiopathic triggers first? It's something to think about.
It really is. Because the answer might completely change your treatment plan.
Right. So stepping back, we aren't just talking about a few random cells surviving in a dish. We have these validated, pure ductal avatars representing genetic, idiopathic, and alcohol backgrounds.
They've built these biological flight simulators and verified the hardware.
Exactly. But the real question is how the internal software of these diseased cells fundamentally differs from healthy tissue, right?
Yes, and that is where the multi-omic profiling comes in. Once the organoids were validated, they ran a PCA.
Principal component analysis, right?
Exactly. They used it to cluster the transcriptomic data, and the separation on the graph was just stark. All of the chronic pancreatitis organoids clustered tightly together, completely separated from the samples of normal adjacent tissue, which they call NAT, and normal pancreas.
Wow, so the transcriptomic landscape of a diseased ductal cell is fundamentally rewired compared to a healthy one?
Completely rewired.
And when you look at the gene volcano plot in the paper, you really see the sheer scale of that rewiring.
Oh, the volcano plot is wild.
Yeah. We're talking about 732 genes significantly upregulated in the diseased tissue. Genes like LINC02582, SLC10082, NOS2, and KLK12.
And simultaneously, 238 genes were significantly downregulated.
Right, so it's not just a slight malfunction. The entire cellular machinery is operating on a completely different set of instructions.
The scale of the dysregulation is massive. But what's fascinating here is the finding that completely shifts our understanding of the disease.
Oh, lay it on me.
The researchers took all of that multi-omic data from the diseased organoids, the thousands of up and down regulated genes, and they ran an unsupervised clustering algorithm on it.
Okay, unsupervised, meaning the computer just looked for patterns without any human bias.
Exactly. They wanted to see how the organoids grouped together based purely on their molecular signatures, completely ignoring the patient's clinical histories.
So it didn't matter if the patient was in the genetic group, the idiopathic group, or the alcohol group?
It didn't matter at all. The algorithm clustered the organoids into 3 distinct molecular subtypes.
Wait, really? Just 3?
Just 3. And those subtypes were entirely independent of the original clinical etiology.
That is insane. So the molecular state of the disease organized itself regardless of what initially triggered the illness.
Exactly. The cause matters less than the molecular state once the disease actually takes hold.
This concept totally shifts the paradigm of how we view chronic conditions. It's think of it like a car engine breaking down.
Okay, I like car analogies.
You might have one car that overheated because it was driven through a desert in the middle of summer.
Yeah.
That's your environmental or alcohol-induced cause. And you might have another car that overheated because a factory robot installed a faulty radiator hose. So that's your genetic mutation.
Right. 2 totally different initial triggers.
Exactly. But when the mechanic opens the hood, both engines are fused together, smoking in the exact same state of catastrophic failure.
They're both just broken.
Right. If the mechanic wants to fix the engines, The history of the desert or the factory doesn't matter anymore. What dictates the repair is the current functional state of the metal.
That analogy perfectly captures the clinical implication here. The etiology initiates the disease, but once it becomes entrenched, the cells fall into one of these distinct functional states.
It really makes you rethink everything.
It does. It strongly suggests that our current clinical trial designs, which often group patients by how they got sick, like separating alcohol patients from genetic ones, might be fundamentally flawed.
Because we should be treating the current molecular profile, not the historical trigger.
Precisely.
Okay, time for our 10-minute mini recap. Bullet 1. PCA mapped the genetic differences between sick and healthy tissue.
Bullet 2. Over 700 genes get turned up, and over 200 get turned down in the disease.
And bullet 3. The disease falls into 3 molecular subtypes, regardless of how the patient originally got sick.
Spot on.
So here's my next question for you listening. If treatments are based on clinical history rather than molecular subtype, how many patients do you think are receiving the wrong class of medication right now?
I shudder to think about it, honestly.
Me too. But if the software is fundamentally rewired into these 3 distinct subtypes, what is the actual physical hardware that breaks to cause all this inflammation?
This is where the researchers zeroed in on CFTR.
Right. We mentioned earlier that 27% of the patients in this biobank had known CFTR mutations. And there's a well-documented link between pancreatitis and cystic fibrosis pathways, right?
Yeah, they actually cite a 2003 paper by Witt regarding that exact connection.
So what does CFTR actually do?
So CFTR, which stands for cystic fibrosis transmembrane conductance regulator, is a vital piece of cellular hardware. It's an ion channel embedded in the surface of the ductal cell. Its primary job is to transport chloride ions out of the cell.
And that creates an osmotic gradient, right? So it pulls water along with it.
Exactly. And this water is absolutely crucial. It flushes the thick, highly reactive digestive enzymes out of the pancreas and into the intestine.
But if that CFTR channel is mutated or jammed?
The fluid doesn't flow. The enzymes stagnate, they activate prematurely, and they essentially begin digesting the organ from the inside out.
Which is terrifying. But the researchers didn't just sequence the DNA to look for these mutations, right? They wanted to see if the channels were physically jammed in these 3D models.
Right. They needed a functional test. So they utilized something called the forskolin-induced swelling assay.
Okay, break that down for me.
It's a highly visual functional readout of CFTR activity. Because these organoids naturally grow as hollow spheres, researchers can suspend them in a gel-like matrigel and provide specific factors like human epidermal growth factor and prostaglandin E2.
Okay, so they're in this nutrient-rich jelly.
Yeah. And then they introduce the chemical forskolin. Forskolin rapidly elevates intracellular CAMP, which is basically a chemical signal that forces the CFTR channels to open fully.
So in a healthy ductal organoid, the CFTR channels fly open, chloride pumps out, water rushes into the hollow center, and if I'm picturing this right, you can physically watch the organoid swell up like a water balloon under the microscope.
Exactly. Rapid visible swelling is the absolute hallmark of healthy transport function.
That is so cool to visualize.
It is. But when they applied forskolin to the chronic pancreatitis avatars with known CFTR mutations, there was virtually no swelling.
Wow.
The channels were physically locked. The transport mechanism was just dead.
Okay, but here's where it gets really interesting. Because the most alarming part of this assay wasn't just confirming that the known pathogenic mutations were broken, right?
Right. They also ran the swelling assay on organoids derived from patients who had CFTR variants that standard genetic screening classifies as non-pathogenic.
Meaning benign, harmless.
Exactly. And those supposedly harmless variants failed the swelling assay, too.
See, this is a critical revelation for diagnostics.
It really is. A standard 2D genetic sequencing test just reads the DNA and cross-references it with a database.
And if the database says a specific variant is non-pathogenic, the clinician just assumes the function is normal.
But this functional profiling in 3D organoids proves that those databases are incomplete. The physical channels in those benign variants were actually functionally impaired.
It really makes you question the absolute authority we give to standard DNA sequencing.
Definitely.
I mean, reading a DNA sequence is like looking at the blueprint of a door. The blueprint might look perfectly fine, right? The structural engineer signs off on it.
Right.
But growing the organoid and running the swelling assay is like actually walking up to the door and turning the doorknob. Sometimes the blueprint is flawless, but the physical door is just rusted shut.
That is exactly what is happening here.
So wait, if a static genetic test tells a patient their variant is harmless, but a living 3D model shows the transport gate is completely jammed, how many invisible structural diseases are we missing right now because we aren't testing the physical mechanics?
We are undoubtedly missing a vast number of functional impairments across all sorts of genetic diseases. It proves why functional profiling, actually watching the live cells attempt to do their job, is superior.
It's mind-blowing. Let's hit our 15-minute mini recap.
Sounds good.
Bullet 1. CFTR mutations jam the cellular transport systems. Bullet 2. The forskolin swelling assay physically proves if a cell's gates are working. And bullet 3. 3D organoids reveal hidden dysfunctions that standard 2D genetic tests completely miss.
Perfect.
So listener, think about this. How would a functional test like this change the way we diagnose other complex diseases like Alzheimer's or autoimmune disorders?
Oh, it could revolutionize them. And that actually sets the stage for the most impactful phase of this study.
The intervention.
Yes. Once they proved the gates were jammed, they introduced CFTR modulators to see if they could physically force them open.
And these modulators are incredible pieces of pharmacology.
They really are. The researchers applied drugs like ivacaftor, elexacaftor, tezacaftor, and lumacaftor directly to the diseased avatars.
And we should clarify, these aren't just painkillers, right?
No, not at all. They are microscopic mechanics designed to bind to the defective CFTR protein and correct its folding, or just force the channel open.
And what happened?
The functional rescue was immediate. When they treated the impaired organoids with these modulators and ran the forskolin assay again, the organoids swelled.
Wow. So the transport function was successfully restored.
It was. But if we connect this to the bigger picture, the true breakthrough wasn't just that they got the water flowing again. It was the downstream biological ripple effect of opening those gates.
Right, because chronic pancreatitis is characterized by massive chronic inflammation and fibrosis. It's not just a plumbing issue. The organ is effectively on fire.
Precisely. And what's fascinating is that these patient-derived organoids naturally retain what the researchers call epithelial intrinsic inflammatory disease programs.
Meaning what, exactly?
Meaning that even when they are isolated in a sterile dish, completely separated from the body's immune system, the ductal cells remember that they are sick.
Oh, that's eerie.
Yeah. They autonomously pump out inflammatory cytokines and mitogenic signals, which are signals that drive erratic cell division and growth.
So the cells themselves are perpetuating the disease state.
Exactly.
But what happened to that intrinsic inflammatory programming when the modulators fixed the CFTR channels?
It plummeted.
Really? Just from fixing the channel?
Yes. By simply restoring the mechanical transport function of CFTR, the researchers observed a significant decrease in both inflammatory signaling and mitogenic signaling.
That is wild.
They didn't apply an anti-inflammatory drug. They didn't apply a drug to stop cell division. They merely fixed the ion channel, and the entire biological storm began to calm itself.
Okay, I love visualizing the cell as a chaotic nightclub.
Oh boy, here we go.
Hear me out. The bouncer at the front door is the CFTR channel. For whatever genetic or idiopathic reason, the bouncer falls asleep.
Okay, I'm with you.
Because the door is completely unmanaged, rowdy guests, which represent the digestive enzymes, the inflammation, the hyperactive cell division, they rush in and completely trash the place.
Yeah.
The club is in pure chaos.
That's a total mess.
But then you give the bouncer a highly targeted cup of coffee. That's the CFTR modulator.
Right.
The bouncer wakes up, stands at the door, and restores normal transport. And as soon as the structural integrity of the door is functioning again, the rowdy guests naturally clear out, the music turns down, and the club's internal environment stabilizes.
You know, it's a funny analogy, but it's a really powerful way to frame it, because it highlights a major flaw in how we historically treat chronic conditions.
How so?
Well, for decades, the medical field has thrown billions of dollars at developing therapeutics to fight inflammation directly.
Right. Anti-inflammatories are everywhere.
But this data suggests that the massive inflammatory response in pancreatitis is a downstream consequence of a mechanical failure. If you just target the inflammation, you're only fighting a symptom.
But if you fix the mechanics, if you wake up the bouncer, the inflammation resolves itself.
Exactly, because the physical stress on the cell has been lifted. The cells basically remembered how to behave properly once the stress was gone.
It fundamentally changes the architecture of chronic disease treatment.
It absolutely does.
Let's do our final mini recap. Bullet 1. CFTR modulators like ivacaftor and elexacaftor successfully restored transport function.
Bullet 2. Fixing this transport calmed down both inflammation and erratic cell growth.
And bullet 3, the disease cells essentially remembered how to behave properly. So listener, if treating the transport mechanism fixes the inflammation, is it possible we've been targeting the wrong end of the disease chain for decades?
It's a huge question.
It really is. So let's pull all this together for you. We started this deep dive talking about a deeply internal, inaccessible organ that leaves doctors guessing.
Right, the muddy waters.
But by extracting that tissue and successfully growing 37 pure ductal epithelial avatars, these researchers have illuminated the black box.
They mathematically mapped the transcriptomic rewiring, discovering those 3 hidden molecular subtypes that completely ignore the patient's original diagnosis.
Right, and they utilized the forskolin swelling assay to physically prove that even supposedly harmless DNA variants were functionally destroying the organ's transport capabilities.
And ultimately, they proved that repairing those broken microscopic gates with modulators instantly dampens the entrenched intrinsic inflammatory programs of the cells.
The clinical relevance of this workflow is just immense. We are entering an era where your treatment is tailored to the functional reality of your unique cells, not just your broad clinical diagnosis.
It really is the end of diagnostic guesswork.
And it leaves us with a truly provocative thought regarding the future of medicine.
Yeah, think about this. If chronic pancreatitis, a disease notoriously difficult to biopsy and treat in living patients, can be decoded by growing these miniature 3D avatars outside the body, what other incurable internal diseases are just waiting for their own organoid biobanks?
Wow.
If your body was inexplicably failing today and you had the choice, would you want your doctor relying on a digital twin made of standard sequencing data or a living, breathing biological twin actually experiencing the disease in a dish?
I mean, when you put it like that,
Yeah,
the biological truth is always found in the living cells.
it always is.
Well, thank you for joining us on this deep dive. Keep asking the big questions, keep questioning the consensus, and we'll see you next time.